All right. Well, first of all, thank you very much for the invitation, Prashant and Chancellor May. It's really an honor to be here and contribute my small portion to that $1 billion of research funding. So to start, I want you to think about Ebola hemorrhagic fever, AIDS, West Nile encephalitis, SARS, bird flu, congenital Zika syndrome, and we can't forget COVID-19. All of these diseases are caused by viruses that emerged by jumping into humans from another animal. And so if this list of diseases doesn't underline the importance of studying viruses, even if they don't cause disease in humans yet, I want you to imagine Ebola-like hemorrhagic fever of unknown origin, a new Zika-like virus that causes birth defects, or a novel coronavirus that causes COVID-32 10 years from now. The virus that causes the next major outbreak is already out there. But imagine if we could actually develop smarter antiviral therapies to be prepared for that next outbreak. Imagine if we could do this in a way that didn't result in drug resistance to these antiviral therapies, the way we have to worry about drug resistance for HIV or influenza virus, or potentially even SARS-CoV-2 with Paxlovid. Now let's get really wild. Imagine if we could have smarter antiviral therapies that would work without even knowing which virus is going to cause the next pandemic. Then we would be truly prepared. And I think that we can accomplish all of that. But to do so, we really have to build generalizable themes about all of these unrelated viruses, but with molecular level resolution. So to explain why we think that molecular details of virus replication are so valuable to combating viral infections, what I want you to do is imagine a little more. Imagine a single cell in a human body and think about what a virus needs to do inside that cell to be successful. I'll argue that it has to do three major things. It has to get into the cell, it has to make more of itself, and then all of those new viruses have to get out. And so viruses have to do all of this with a very small set of viral machinery. And so compared to the host, which has a much larger set of molecular machinery, viruses have started to hijack the molecular machinery of the host. Viruses are master manipulators of cells, and they use the molecular machinery of cells to turn cells into tiny little viral factories. And so instead of targeting the viral molecular machinery with our antiviral therapy, imagine if we instead attacked the host molecular machinery. And this is exactly what my team of chemical engineers and biologists really think about when we do all of our experiments. So my team builds new systems to study these molecular events of virus replication with quantitative detail. And I could talk for hours about how we do all of this work, but what I really want to do today is tell you about why this work is so important and why it's powerful through the lens of building smarter antiviral therapies. One of the types of molecular events that my group really loves to study are protein interactions between a viral protein and a host protein. So sometimes these protein interactions help the virus replicate. Sometimes these protein interactions actually help the host defend itself against the virus. Sometimes these protein interactions actually cause disease on a macroscopic scale, like we've discovered with the birth defects that are caused by Zika virus infection. And so how does studying these molecular interactions actually help us build smarter therapies? Well, if you think about how your traditional antiviral drug is developed, it's developed by targeting a viral protein. And so the drug has to really fit really nicely with the shape of the viral protein to be effective. Slight changes in the shape of that viral protein could render that drug completely useless. These differences in shape could be because you're trying to use the drug on one virus, even though it was designed for a completely different virus. So we've got Paxlovid now, but what if there's a novel coronavirus 10 years from now? Will Paxlovid still work? Will that viral protein still look close enough to SARS-CoV-2's viral protein? We don't know. The other way these differences in protein shape can come about is through the actual mutation process that occurs during virus replication. And that's exactly what we see with drug-resistant strains of HIV or other viruses. So now let's flip this around. Let's actually target the host machinery instead. One of the advantages of targeting the host machinery is that now mutations in the viral proteins don't really matter anymore. The drug will still bind the host machinery quite well. And so for a virus to really develop resistance to a host-targeted therapy, the virus would have to actually find a replacement host target. That's a much more difficult problem for the virus to solve. And so this therapy effectively traps the virus where it is. Another exciting advantage of a host-targeted therapy is that actually a lot of unrelated viruses will still use the same host machinery to replicate themselves. And so if you can identify that machinery across many different viruses, you could potentially have a drug that works against SARS-CoV-2 and Ebola virus and Zika virus. And so differences in the viral protein shape don't really matter anymore. What are the implications of this? Well, let's suppose we had a drug that was active against SARS-CoV-2, Ebola virus, and Zika virus. Well, there's a high probability that that host-targeted therapy would also be active against a novel coronavirus that we identify 10 years from now, or a new Zika-like virus that causes birth defects. And so this is just one way we think that we can build smarter antiviral therapies through targeting virus-host protein interactions. Now, what's stopping us from this being a reality? Well, first of all, we just need more data. There is such a rich world of viruses out there that we know very little about. We don't understand how they interact at the molecular level with the host machinery. So we can start to fill these gaps in knowledge with unbiased, discovery-based research. And we can focus our efforts on virus families that have a high risk for emergence and new outbreaks. The tricky part, though, is that federal funding agencies don't always fund this type of discovery science or fishing expeditions to the level that you would need to support the type of vision I just described. So we really do need to find new ways to support this type of discovery research. The other thing that we need to do is build new partnerships. We need to identify collaborators with complementary skill sets that can help us identify the virus families that pose the highest risk, that can test the host-targeted therapies once we identify all of those host targets. So all of that capability actually exists here on UC Davis campus, which is really great for me because this is my vision. But these are just a few of the pieces of the puzzle that we need to lay down to make this a reality. But it's a start for building smarter antiviral therapies. Thank you.